Hello, everyone. We are Design Team 9, and our project involved designing and modeling an aquaponics system for a rural village in Uganda. Our presentation today will cover aspects of bioreactions, the nitrogen cycle, and the modeling, the ramp-up time of our system until it reaches steady state. We have already covered some aspects of biological reactions in kinetics class, but our presentation today addresses the conditions before a system reaches steady state, which goes beyond what we have already covered in class.
For this tutorial, we recommend that you open the provided worksheet and download and open the skeleton code provided in MatLab. We intend that you will be able to fill out the questions on the worksheet in order as you follow along. Also, there will be places where we ask you to pause the video to complete calculations and fill in the skeleton MATLAB code provided with your calculated values.
To begin, we can review some types of kinetics that we have already learned. First, we have the elementary reactions. This is the simplest type of kinetics and can be characterized by reactions where the reactants have a direct and proportional effect on the product's concentration. The rate equation is defined by the concentrations of the reactants and a rate constant K. Another type of reaction kinetics we've learned about is Michaelis-Menten kinetics. What makes this different from elementary reactions is that an enzyme is present. The enzyme acts as a catalyst where it's involved in the reaction but is not consumed or produced. This changes the rate equation to include the initial concentration of the enzyme and also an additional constant called the Michaelis-Menten constant. So now let's consider the kinetics of a reaction that combines characteristics of both of these concepts. In Monod Kinetics, cells act as a catalyst in the reaction but also act as a product. This combines concepts from both elementary and Michaelis-Menten kinetics, as the cells are proportionally produced in relation to the amount of reactant present, but they are also necessary for the reaction to move forward. In the rate equation for Monod Kinetics, the setup is very similar to Michaelis-Menten. The main difference, however, is that we use a Monod constant instead of the Michaelis-Menten constant. The Monod constant refers to a substrate saturation constant, which is defined by the concentration of A, where the cells reproduce at half of their maximum rate. Since the cells are living organisms, their behavior can differ from regular non-living products. And since the concentration of cells can affect the rate of reaction, it's important to understand their growth trends. The general growth curve of the cells can be defined by 4 phases. The first phase is the lag phase. During this time, the initial concentration of cells is relatively constant. This is because the cells may require time to mature and adapt to the growth conditions before they are ready to divide and produce more cells. The next phase is the exponential growth phase. At this stage, the cells are actively growing and producing as they consume and convert the reactants, which act as food for the cells. Next is the stationary phase. This occurs when the reactant or food concentration for the cells is constant. Here, the rate of growth and rate of death of the cells are equal, so the population size remains constant. The last phase refers to cell death. At this point, the food for the cells have run out and no more cell growth is able to occur. Because of this, the population will decrease until all the cells die or more reactant is added to the system.
Presented here are 3 equations that are crucial in modeling the growth rate of bacteria and the change of concentration in the substrate. Equation 1 is attributed to Monod and relates the maximum specific growth rate of a bacterial species to the substrate concentration. Umax is the maximum specific growth rate of the species, S is the growth substrate concentration, and Ks is the substrate concentration when U is at half of its maximum value. The second equation is a general equation relating the mass of new cells to the mass of substrate consumed. This is defined as the yield coefficient. The second equation describes the specific growth rate of the cells. It is defined as the mass of new cells produced divided by the original mass of cells times the time over which these cells were produced. These general equations will be combined with the Monod equation to yield specific equations describing the important parts of the system. Combining equations 1 and 3 yields an equation 4, which is useful in describing the time rate of change of the biomass. This will show how much new biomass is produced per unit time. This is an important equation because it describes the amount of new biomass of each species produced per unit time over the growth phase of the bacteria. This is important because it will allow us to determine when the bacteria colonies will be fully grown and when we can add the rest of the components to the aquaponic system. In a similar manner, combining equations 1 and 2 yields equation 5, which is useful in describing the rate of substrate utilization. This will also allow us to see the rate of consumption of the compounds the bacteria use to multiply. Similar to the other equation, this will allow us to determine when the system reaches a state where the consumption of the substrate is at a steady value.
To understand why these biological reactions are important for our system, we will briefly review the nitrogen cycle. Nitrogen is an essential element for the growth and development of plants. Nitrogen is present in the soil in the form of NH3, which cannot be readily used. A series of nitrification reactions must take place in order for the ammonia to be converted into useful compounds for the plants. Nitrifying bacteria in the soil convert this ammonia first into nitrites. These nitrite compounds are then taken in by a different strain of nitrifying bacteria and converted into nitrates, which are readily used by plants to grow. Because bacteria are required to convert the nitrogen into a usable form, the entire nitrogen cycle depends on the nitrifying bacteria. The nitrogen cycle in our fish tank, in our system, starts in the fish tank. Waste produced by the fish is the primary source of nitrogen in the form of ammonia. This dissolved ammonia is again not able to be used by the plants directly. The water exits the fish tank and moves to a series of filters. One of these filters is a biofilter. The purpose of this filter is to house and grow nitrifying bacteria to convert the ammonia into usable nitrates for the plants. The bacteria have two primary functions. First, they convert the harmful ammonia to prevent buildup. A buildup of ammonia would be deadly to the fish and the plants. Secondly, they produce the necessary nitrates that allow for the growth of the plants. The nitrate-rich water then leaves the series of filters where it is brought to the plant beds. The plants then uptake the nitrates from the water, and the water is returned to the fish tank where the cycle starts again. As our modeling will show, the ammonia and nitrite levels will decline, and the nitrate levels in the system will reach a steady state value. This means that the colonies of bacteria have grown to meet the conversion demand of the system, and the fish and plants can be added. It is important to note that the bacteria need to be grown before the fish and plants are added. If the fish and plants are added before the colonies are grown, the ammonia levels will rise rapidly and be deadly to the system.
In this animation, you can see that fish waste is produced as ammonia, which is then converted to nitrite followed by nitrate. This nitrate is then pumped to the plants where it can be used as food for growth. The filtered water, free of nitrates, is returned back to the fish tank to complete the nitrogen cycle.
The two strains of nitrifying bacteria that are present in our system are Asynchronous and Bioreactor. Both will be grown in large quantities in the biofilter. Asynchronous is the first step in the process. It converts the ammonia from the fish tank into nitrite compounds. The second strain, Bioreactor, then converts the nitrite compounds into usable nitrate compounds.
The bacteria pictured here is a Asynchronous and is used to oxidize ammonia to nitrite. The bacteria pictured here is a Nitrobacter and is used to oxidize nitrite to nitrate.
Here are the general reaction equations for Nitrosomonas and Nitrobacter. The aquaponic system is constantly dependent on the nitrifying bacteria to convert the harmful ammonia into usable compounds for the plants. The system must be maintained at certain conditions in order for the bacteria to adequately convert the ammonia into nitrates. These bacteria thrive at temperatures ranging from 17 to 34 degrees Celsius. Temperatures higher or lower will negatively influence the growth and conversion rate of the bacteria. Similarly, these bacteria function best at a water pH of 7.2 to 7.8. Factors such as temperature, pH, and concentration of the reactants can all have an effect on the conversion rate of the bacteria.
The first equation listed here represents the Nitrosomonas growth rate, while the second represents the Nitrobacter growth rate. In these equations, K represents the growth rate constant for each respective bacteria species. Cm and Cb are the concentrations of the respective bacteria. Lowercase x is the concentration of ammonia. Lowercase y is the concentration of nitrite. Uppercase X is the saturation constant of ammonia, and uppercase Y is the saturation constant of nitrite. We've included the parameters and their meaning in a table on the worksheet, so refer to that if you ever are mixed up. It is important to note here that the Nitrosomona concentration is dependent on the concentration of ammonia, X, which also changes as a function of time. Similarly, the Nitrobacter concentration is dependent on the concentration of nitrite, Y, which also varies over time.
The equations that model the concentration of each bacteria were solved as shown here for Nitrosomonas and Nitrobacter respectively. Lowercase x and lowercase y are the ammonia and nitrite concentrations mentioned previously. EM and EB are dry mass constants, and FM is the conversion factor from ammonia to nitrite, which is assumed to be 99%.
We substituted the bacteria concentration expressions from the previous slide into the growth rate equations to yield the following. Since these expressions are a function of bacteria concentration, ammonia concentration, and nitrate concentration, we need to simplify further to have a single expression for ammonia and nitrite, not in terms of bacteria concentration.
The derivative of the expressions for the concentration of Nitrosomonas and Nitrobacter were taken with respect to time to achieve a relationship between the concentration of bacteria, which is represented as dCm/dt and dCb/dt here, with respect to time, and the concentration of ammonia and nitrate with respect to time, seen here as dx/dt and dy/dt. As you can see, the concentration of Nitrosomonas is only a function of the concentration of ammonia, represented by dx/dt. However, the concentration of Nitrobacter is a function of both the concentration of ammonia and nitrite, the latter being dy/dt. This indicates that we will need to use a numerical solver to obtain the concentration of Nitrobacter with respect to time.
By replacing the dCm/dt and dCb/dt terms with expressions for dx/dt and dy/dt and simplifying, we now have equations to model the concentration of ammonia and nitrite in the system over time as seen here. These are the final equations that we will be using in MatLab. As can be seen in the equation, the concentration of ammonia over time is a function of several of the constants previously mentioned, along with the concentration of ammonia, lowercase x. As for the concentration of nitrate over time, as seen on the bottom of the page, it too is a function of several of the constants described. That being said, this equation is also a function of ammonia concentration, lowercase x, nitrate concentration, lowercase y, and the ammonia rate, dx/dt. For the MATLAB code used, the dx/dt term expression was plugged into the dy/dt expression. Overall, we now have two rates, the second of which is dependent on lowercase x and y, or ammonia and nitrite concentration, respectively.
The growth rate and saturation constants in the previous equations are temperature dependent. The equations shown here depict a logarithmic relationship between the constants and the temperature, all of which were determined from a linear regression of experimental data in Knowles et al. For Nitrosomonas, there is an observed 9.5% increase in growth rate constant for every increase in degrees Celsius. For Nitrobacter, there is a 5.9% increase. For Nitrosomonas, there is an observed 11.8% increase in the saturation constant for every increase in degrees Celsius. For Nitrobacter, there is a 14.5% increase. Please pause the video now and fill in the table on the worksheet with your calculated values for Km, Kb, capital X, and capital Y.
Please open the provided skeleton MATLAB code for modeling the startup of a bioreactor in an aquaponic system. You will need your calculated values of Km, Kb, capital X, and capital Y from the table in the worksheet. In the code, the array V holds the constants for our 4 rate equations. The array Y0 holds the initial concentrations of ammonia, nitrite, and the 2 bacteria species. X represents the time domain. The final line calls the implicit Euler solver and stores the calculated values in the matrices X out and Y out.
We calculated the residual and the Jacobian, where the first row represents the concentration of ammonia, the second row represents the concentration of nitrite, the third row represents Nitrosomonas growth, and the fourth row represents the Nitrobacter growth. The last row of the residual function calculates the residual per the definition of residual. The rows of the Jacobian represent the same equations as the residual. For example, M12 represents the matrix location of the first row in the second column. The first column of the Jacobian represents the derivative with respect to the concentration of ammonia, lowercase x. The second column of the Jacobian represents the derivative with respect to the concentration of nitrate, which is lowercase y. The third column of the Jacobian represents the derivative with respect to the concentration of Nitrosomonas, Cm. The fourth column of the Jacobian represents the derivative with respect to the concentration of Nitrobacter, Cb.
To calculate the concentration of nitrate, we created a loop that calculates the Riemann sum to approximate the integral of the nitrite curve. We assumed a 99% conversion of nitrite to nitrate. When the concentration of nitrite decreases to 0, the else condition dictates that the concentration of nitrate is equal to the maximum concentration of nitrate.
Enter your calculated values of Km, Kb, capital X, and capital Y into the V array. Run the code to generate the desired graphs. Make sure to publish the code as a PDF.
Pictured are the 2 graphs that you should have generated from your MATLAB code. You can use these to answer the remaining questions on the worksheet. On the first graph, we see the aquaponics biofilter startup nutrient concentrations of ammonia, nitrite, and nitrate. The ammonia concentration starts at 1.5 milligrams per liter, since this is the nutrient initially added to the system, which would either be in the form of fish food or fish waste in an aquaponics system. As soon as ammonia is added, though, the Nitrosomonas oxidize it to produce nitrite. This is seen as the orange curve that begins increasing starting at time equals 0. Slightly after time equals 0, the yellow curve representing nitrate begins to increase as well. This is because the nitrite produced from the ammonia is also oxidized by Nitrobacter into nitrate. After approximately 2.5 days in the modeled system, the nitrate concentration reaches its peak concentration and plateaus. Also around this time, the ammonia and nitrite concentrations have become 0. At these conditions, the toxic ammonia and nitrate have been converted in the system to nitrate, which is able to be used by the plants. Therefore, at this time, the system is ready for startup and fish can now be added.
From the second graph generated, you can see how the concentration of Nitrosomonas and Nitrobacter bacteria change with respect to time. We selected a standard initial concentration of Nitrosomonas and Nitrobacter bacteria to populate our biological reactor. You can see from the graph that the concentration of Nitrosomonas increases to a maximum of 0.11 milligrams per liter around the second day. Nitrobacter increases more slowly and reaches its maximum of 0.09 milligrams per liter around the third day. The concentration of Nitrobacter peaks after Nitrosomonas because, as stated earlier, the concentration of Nitrobacter with respect to time is dependent upon both the concentration of ammonia, little x, and the concentration of nitrite, little y. However, nitrite cannot be produced until the nitrosomonas oxidize the ammonia, delaying the increase in concentration of Nitrobacter. Because in this simulation only a set initial concentration of ammonia was added to the system, both the concentration of nitrosomonas and Nitrobacter go to zero once all of the ammonia and nitrite are used. In a steady-state system, such as our established aquaponic system, there would be a steady introduction of ammonia. This would result in the nitrosomonas and Nitrobacter reaching a new steady-state equilibrium greater than zero. Once you have completed the questions, submit the worksheet in a published MATLAB PDF with the desired graphs.